Okay, today we're going to be discussing cell-to-cell interactions. Now, every living organism has to respond to their environment, right? If you're a living organism, you have to respond to both internal and external environmental changes. In addition, we have to be able to cooperate and communicate with nearby cells. How are we going to do this? How are we going to interact with other cells? That's going to take place at the level of the cell's surface. So we know that we already have our plasma membrane, right? Our plasma membrane is that phospholipid bilayer, has embedded proteins in it, so we call it the fluid mosaic model. And we understand that the plasma membrane is going to contain all of our internal structures of the cell, right? By creating that environment inside the cell. However, surrounding our plasma membrane, we're going to have an extracellular surface. Our extracellular surface is going to provide an additional protective layer, help define the cell shape, as well as attach that one cell to another cell. So plasma membrane is good, but surrounding our plasma membrane, we have to have our extracellular surface. Why? Your extracellular surface is important for providing and defining that cell shape, giving it the abilities to attach to another cell, and also acting as that first line of defense. So what makes up our extracellular surface is a fiber composite, our extracellular matrix. Our extracellular matrix is made up of different proteins and sugars. All those fibers involved in our extracellular matrix is also going to help resist tension. All right, here's our extracellular matrix. So over here we see our plasma membrane. Directly outside of our plasma membrane, we're going to have our extracellular matrix. Our extracellular matrix is going to be a web of proteins and carbohydrates directly at the cell surface. The fibrous component of our extracellular matrix is collagen. Collagen is a form of protein. Our collagen is going to bind together to form these complex fibers, therefore forming a flexible extracellular layer. Ground substance of our extracellular matrix is going to be made up of our proteoglycans. Proteoglycans are important because they're hydrophilic. They're going to therefore attract water and then form a gel. So far we need to understand that our cell is going to have an internal environment. As a living organism, we want to make sure that this is an equilibrium homeostatic environment compared to our external environment. So living organisms are constantly responding to internal and external changes. How are we able to do so? It's due to the fact that we have this extracellular surface surrounding our plasma membrane. Our plasma membrane is going to create an environment inside the cell. Our extracellular matrix is directly outside of that plasma membrane. It's composed of different proteins and sugars. Our major fiber is collagen. Collagen is going to form cross-links and form an overall extracellular layer that is flexible. We also have a ground substance. Our ground substance is going to attract water. Our ground substance is made up of the proteoglycans. Proteoglycans are hydrophilic, therefore they're going to attract water and form a gel. So, different tissues of our body will have various different amounts of our extracellular matrix. So, cells that have a similar function are going to combine, and that's going to form a tissue. So a tissue is essentially similar cells are going to function as a single unit. The amount of extracellular matrix is going to differ in each tissue. So for example, our lungs, right? Our lungs constantly have to expand and then recoil, right? That's how breathing works, essentially. So because our lungs are constantly expanding and then recoiling, they use the protein elastin in their extracellular matrix. That elastin protein is going to allow the lungs to stretch and then recoil, with every kind of respiration. In addition, our extracellular matrix is going to have these cross-linking proteins. Our cross-linking proteins are laminins that are going to bind to our integrins that are located on the cell surface. So your extracellular matrix has laminins. Integrins are on the cell surface. Laminins will bind to integrins on the cell surface to make it like one unit. So these cross-linking proteins are going to help attach the extracellular matrix to the plasma membrane. Why? The plasma membrane has the integrins. Our extracellular matrix has the laminins. Integrins are also going to anchor the cytoskeleton to the extracellular matrix. Alright, plants. Most plant cells are going to be surrounded by a cell wall. When a new plant cell forms, they're going to secrete a fiber composite called a primary cell wall. Primary cell wall is going to be composed of very long cellulose strands. They're going to get all bundled up and form a microfibril. Between our microfibrils, we're going to have polysaccharides such as pectin. Now, pectin is going to help keep our cell wall moist. Why? Because pectin is going to attract water. Once again, that is hydrophilic. So this is similar to the extracellular matrix. In our extracellular matrix, we had our proteoglycans attracting water, forming a gel. In our primary cell wall, we're going to have pectin that is attracting water, forming like a gel-like substance and help keeping that cell wall moist. All right, more about our primary cell wall. The primary cell wall is going to help define the shape of the plant cell. Once again, this is similar to our extracellular matrix. The primary cell wall will counteract the turgor pressure that is experienced by that large central vacuole. Turgor pressure is very important for young plants. Why? It's going to help secrete expansins. Expansins, therefore, are going to disrupt cross-linking between these microfibrils, and that's overall going to allow the cell to grow. We can also have a secondary plant cell wall. The secondary cell wall is going to take place between the plasma membrane and the primary cell wall. Our secondary cell wall is an important structure because its structure is related to that particular cell's function. So if we're a secondary cell wall in a leaf cell, we're going to contain waxes. If we're talking about a secondary cell wall in wood, we're going to be talking about containing the protein lignin. So if a tissue is composed of multiple cells, we have to have a way to kind of link these cells together. So the basis of multicellularity is due to physical connections between the cells. It's going to help maintain structure and functions of tissues, right? A tissue is composed of multiple cells. In order to form a tissue, we have to link these individual cells together. The different structures that hold these cells together are going to differ between organisms. So first, let's talk about our plant cells. What I want you to understand about plant cells is you definitely have to understand the idea of having a primary cell wall, understand the idea of that central vacuole in terms of turgor pressure. So now we have two separate plant cells, right? And I want to form a big plant. In order to form that large structure, I have to link together these individual plant cells to one another. So I'm going to take one plant cell, right? And this plant cell is going to have a primary cell wall. And I want to attach it directly to another plant cell, and that other plant cell is also going to have a primary cell wall. We're going to glue this together with a substance called the middle lamella. So plant cells will be glued together by the middle lamella. The middle lamella is going to be continuous with adjacent plant cells' primary cell walls and is comprised of pectins. So here we have our plant cell wall, our plant cell. We're going to have our primary cell wall get directly attached to another primary cell wall. How is this primary cell wall glued to this one? Through the middle lamella. So the primary cell wall is this light green dotted one. This is the primary cell wall of one cell. Here's the primary cell wall of another cell. These cells have to come together. We're going to fuse the primary cell wall utilizing our middle lamella, and that's how these cells are going to form a connection. In animal cells, it's a little different. In animal cells, we're going to discuss tight junctions and desmosomes. We're also going to talk a little bit about our gap junctions. So gap junctions are a little different because it's going to involve a direct connection between the cytoplasm of two adjacent cells. Gap junctions are very important when we have to talk about coordinating activity. So a good example would be our heart, right? We want all the cells of our heart to function at the exact same time, right? You don't want the left portion of your heart beating and then a minute later the right side of your heart beating, right? It has to be a synchronous activity. That's when our gap junctions are important. So gap junctions are going to involve a direct connection between cytoplasms of cells, therefore allowing for synchronous activity. All right, tight junctions. Tight junctions are going to be tight. So tight junctions are composed of membrane proteins in adjacent animal cells, right? So two animal cells are directly next to one another. The proteins are going to line up and bind to each other, stitch these two cells together, and help create a watertight seal, right? It's a tight junction. It's going to be a watertight seal. Tight junctions are going to be variable and dynamic though. Although they're tight, they can open and close, and depending on a tissue, it may be looser, right? You can have a tight junction that may be looser in one tissue compared to another tissue. Ultimately, our tight junctions are going to limit the passage of ions and molecules through the space between cells. All right, so here's our tight junction. So this would be one cell, right? You see our plasma membrane. This would be a cell directly next to it, right? An adjacent cell. This would be your tight junction. The membrane proteins are going to bind to one another on the cell wall, on the cell membrane, and form that tight junction. Tight junction, think of a watertight seal. We're not allowing material to pass through. Desmosomes. So desmosomes are made up of proteins that link the cytoskeleton of adjacent cells. Now we're talking about the cytoskeleton. Therefore, we're going to start talking about resisting pulling and the shearing forces that can act on a cell. Your desmosomes are going to be common in our epithelial and muscle tissue. All right. Discovery of cell-to-cell adhesion proteins. So researchers hypothesized that different cell types are going to have different adhesion proteins. Only cells with the same or complementary adhesion proteins can attach to one another and eventually form a tissue, right? I'm not going to have heart tissue in my lung tissue. They have different functions. They're going to be made up of different fibers. It can't happen. So cell-to-cell adhesion proteins, in order to start linking cells together, they have to be specific to one another. They have to have the similar adhesion proteins or complementary adhesion proteins to link to one another. So researchers explore this idea using antibodies. Antibodies are essentially proteins that can only bind to a specific protein. So antibody A will only bind to protein A. By utilizing antibodies, we may change the other protein structure or interfere with its inability to interact with other molecules. If an antibody is going to prevent a cell from adhering, the antibody is probably going to be bound to an adhesion protein. So some major classes are cadherins. Cadherins are the attachment molecules in our desmosomes. Remember, desmosomes are important for linking together our cytoskeleton. Each cadherin binds only to cadherins of the same type. This trait is allowing cells of the same type to attach specifically to each other. So a cadherin A will only bind and interact with a cadherin A of another cell. Cadherin A will never mingle with cadherin B. They will never form that connection. Okay, so it is through our desmosomes and our tight junctions, that is how we connect cells to another cell. Well, one cell to another cell. Gap junctions are important for cell communication between cells. How? Gap junctions have that gap. So direct connections between cells in the same tissue is going to allow a cell to work together as a unit, right? If you link two lung cells together, eventually we're going to form that lung tissue and it can function as a whole. So ions and small molecules will be able to pass between cells. This can have an impact on regulating gene expression, and it can activate or inactivate other proteins involved in that reaction. Once again, these are called gap junctions in animal cells. So gap junctions are important because they allow for the direct communication, direct connection between cytoplasm of two adjacent cells. Having that gap is going to allow the flow of small molecules between cells, ultimately making sure that we're synchronizing cellular activity. Alright, so here are little gap junctions between two adjacent cells, where we see one plasma membrane and another plasma membrane. Our gap junctions are membrane proteins in adjacent cells that are going to line up together and form a channel in order for small ions and molecules to pass between the cytoplasm. Plant cells' gap junctions, I say that in quotes, are going to be called plasmodesmata. So plant cells do not have true gap junctions, instead they have plasmodesmata. Plant cells are connected by gaps in their cell walls through plasmodesmata. Plasma membranes, cytoplasm, and a smoothie R, the two cells, are therefore going to connect through plasmodesmata. Plasma membrane is then going to separate plant tissues into our symplast, which involves our shared cytoplasm, and our apoplast, which is our extracellular space. Okay, so now let's talk about cell-to-cell signaling in a very large multicellular organism. So distant cells have to be able to communicate with one another. How are they going to communicate? Through cell signaling. Cell signaling is going to involve the use of neurotransmitters and hormones. So neurotransmitters may open or close channels in distant cells. Hormones are information-carrying molecules. Hormones are secreted from the cell. Okay, so what are hormones? Hormones are typically going to be very small molecules. They could be a peptide, a steroid, or even a gas. Hormones are usually in a very small concentration, but they will have a large impact on the activity of its target cell. So on its target cell, we're going to have a signal receptor. Our hormone will bind to that signal receptor. It may change its shape and activity after binding to a hormone, the target cell itself. Only cells with an appropriate signal receptor will respond to a particular signaling molecule. Right? Our receptors are going to be very specific in who they want to talk to, who they want to communicate and bind with. In order to fit into that receptor, you have to have the right amino acids, the right orientation, in order to bind with that receptor. So a hormone will only exert its effect if it can bind to its appropriate signal receptor on the cell membrane. So hormones may be soluble or insoluble in lipids. Our lipid-soluble hormones are able to diffuse directly across our plasma membrane. Remember that our plasma membrane is composed of phospholipids. These lipids are hydrophobic. What can pass through a hydrophobic material? Something else that is also hydrophobic, such as a lipid-soluble hormone. So our lipid-soluble hormone is able to directly diffuse through our plasma membrane and then enter the cytoplasm of the cell. Once inside the cell, it will bind to receptors inside of the cytoplasm and change cell activity directly. How is it able to change cell activity directly? The hormone receptor complex that is formed in the cytoplasm is going to get transported to the nucleus where it alters gene expression. So, hormone signaling could either be soluble or insoluble in terms of lipid solubility. If we have a fatty hormone, like usually a steroid-based hormone, why? Because steroid is like 4-ring structure, like cholesterol, very fatty, can pass through our membrane. It's going to pass directly into our cytoplasm. In the cytoplasm, it's going to bind to its specific receptor. So now we're going to have a steroid interacting with its specific receptor. Once we have this hormone receptor complex, this hormone receptor complex is going to get transported to the nucleus, where it will directly alter a target gene DNA. Okay, now let's discuss our lipid insoluble signaling molecules. Now, our lipid insoluble hormones are unable to pass through our plasma membrane. Why? They are insoluble in lipids. What is our cell membrane made up of? Phospholipids. So, lipid insoluble hormones are unable to pass directly through. Lipid insoluble and large hormones do not cross through the plasma membrane. Hormones that cannot diffuse across the plasma membrane have to bind to external receptors on the cell membrane and then induce a second messenger signaling cascade system. So, when an insoluble lipid signal binds to a cell surface receptor, its binding is going to trigger a signal transduction cascade, which is going to translate into a bunch of intracellular signaling molecules, our secondary messenger systems. Then we will convert that extracellular hormone into an intracellular signal, which can then impact our DNA. So lipid insoluble is unable to pass through our plasma membrane. What's going to happen instead is that insoluble molecule will bind to a cell surface receptor. Once that hormone or molecule binds to a receptor, it's going to induce intracellular signaling molecules. So we went from an extracellular message, translated it into an intracellular message. That's our signal transduction. Then that secondary messenger will impact our DNA in the nucleus. So the message that is going to be transmitted by the hormone is essentially going to be amplified in the cell. I mentioned before that hormones are in low concentrations. This signal transduction system is going to amplify that tiny concentration of hormones. We have different methods in how we can amplify the signal. It could be a signal transduction cascade in which many ion channels are going to get opened. It could be activated or amplified through a G protein coupled receptor, or it could be through an enzyme linked receptor. So let's first talk about our G protein receptors. So G proteins are intracellular peripheral membrane proteins. So let's break this down. They're intracellular on the periphery of a membrane. So here we see our phospholipid bilayer, we see our receptor. Insoluble molecular hormone has to bind to that receptor. Once it binds to the receptor, we can activate a G protein. The G protein is an intracellular, so we're inside the cell, periphery membrane protein, right? We're on the periphery of our cell membrane, but we're inside the cell, we're not on the outside. When G proteins are activated by their signal receptor, we're going to trigger production of a second messenger. The second messenger is going to be a small, non-protein signaling molecule or ion. The extracellular signal is therefore going to produce an intracellular signal. So, once again, We have our receptor. Directly attached to our receptor is this G protein. G protein is an intracellular peripheral membrane protein. When our insoluble hormone binds to our receptor, this G protein is going to become activated. Once activated, we're going to trigger second messenger cascade. Our second messengers are going to be small non-protein signaling molecules or ions. That's how we go from an extracellular message to an intracellular message. Alright, more about our G proteins. G proteins are regulated through guanine nucleotides. When a G protein is bound to GTP or guanosine triphosphate, its shape is going to be altered and is activated. I like to think of this as ATP is our major energy source. We can't do anything without ATP. Think the same for GTP. GTP is like another energy source. It's going to activate that G protein. Your G protein will only be active if it is bound to GTP. So when a phosphate group is removed from the bound GTP to form GDP, the G protein is inactivated. So if GDP is bound to our intracellular G protein, it's inactive. If GTP is bound, right, similar to ATP, it's going to be active. Only when GTP is bound will we convert that extracellular signal into an intracellular signal to produce second messengers. If we do not have an active G protein, we will not be able to form these second messengers. So G protein coupled signaling is going to involve three major steps. First, we're going to have a signaling molecule bind to a membrane-bound receptor. Remember the whole idea with G protein coupled receptor, we need that membrane-bound receptor because these hormones, these molecules are unable to pass through that lipid bilayer. Our associated G protein is going to exchange a GDP for GTP, right? GTP is the only active form. The G protein will only work if it has GTP bound. The active G protein subunit will then go on to activate a nearby membrane enzyme that is going to catalyze the production of a second messenger. So let me show you what that means. So our signal bound to our receptor, G protein is directly attached to the receptor. We see here this is our inactive G protein form. Why? It has GDP bound. Once we form our signal receptor complex, our G subunit is going to split into two. That's when it's active because one is going to have a GTP bound. The GTP bound subunit of our G protein is going to travel along the length of our plasma membrane and bind to a specific enzyme. Once that active GTP binds to that enzyme, we're going to trigger the release and production of our second messengers. That second messenger is going to be how we go from an extracellular signal to an intracellular signal. Remember that cannot take place unless you split the G protein in half and change from GDP to GTP. Alright, so what are these second messengers? Second messengers are important because they're going to be small molecules, and these smaller molecules can diffuse rapidly throughout the cell. Another good benefit about our second messengers is that they're produced quickly and in large quantities. Therefore, they're good to amplify that hormone signal. Hormones are always produced in very low quantities. Why hormones still work the way they do is through our second messengers. So no second messenger production would mean no response of that hormone. Now, some of these second messengers can activate protein kinases. Kinases are enzymes that add phosphates to other proteins. Usually when we add a phosphate, we're either activating that protein or deactivating that protein by changing its structure. So a kinase is an enzyme that adds a phosphate group to other proteins. By Well, through addition of that phosphate, we can either activate or inactivate that protein. So here are just some examples of second messengers. We have our inositol triphosphate, our IP3, cyclic guanosine monophosphate, CGMP, our calcium ion. So just be familiar that they're small. Alright. Now let's talk about signal transduction through enzyme-linked receptors. So our enzyme-linked receptors are transmembrane proteins. These transmembrane proteins are going to bind a hormone signal and then directly catalyze a reaction inside the cell. The best known enzyme-linked receptor we studied is our receptor tyrosine kinases, or our RTKs. So receptor tyrosine kinase signal transduction is going to involve 5 major steps. The first step is when our hormone binds to our 2 RTK subunits and then causes them to form a dimer. Once we form that dimer, the RTK is going to get phosphorylated by ATP. Proteins will then build a bridge between RTK and the RAS protein, therefore activating RAS. An activated RAS subunit will then phosphorylate a protein kinase. Once our protein kinase is phosphorylated, this is going to trigger a phosphorylation cascade for protein kinase. So now we're going to phosphorylate everything. We begin with 2 receptor tyrosine kinases. A signaling molecule, some hormone, is going to bind. Once that hormone binds, we're going to form an RTK dimer. Now, an RTK, it's a kinase. It has the ability to phosphorylate. It will only phosphorylate itself once we form a dimer. We only form a dimer when our signaling molecule binds. Okay, signaling molecule has bound, right? We formed our dimer. Once we form a dimer, we're going to activate our RTK kinase activity. So now, that's why we see these little phosphates here. The RTK phosphorylated itself. Once our dimer is phosphorylated, it's going to bind other proteins in the area. One of the special important proteins you have to know is going to be our RAS protein. Once RAS binds through binding proteins, it's going to exchange GDP for GTP. Once RAS has a GTP attached to it, we're going to have a phosphorylation cascade. And that's what we see here. Our activated RAS, it's activated because it has a GTP bound, is going to trigger phosphorylation and activation of a protein kinase. Alright. Very dependent on signal to signal as well as from cell to cell. Our signal response therefore falls into 2 different categories. A change in which the genes are being expressed in the target cell, or a change in activation or deactivation of a particular target protein that already exists. So signal deactivation. Signal and deactivation takes place when we turn off a cell's signal. So turning off a cell is just as important as turning on a cell, right? So cells are going to have these automatic and rapid mechanisms for a signal deactivation. We said before that kinases were enzymes that add a phosphate. Usually when we add a phosphate, we're activating something. Phosphatases is a specific enzyme that removes a phosphate. So a phosphatase is an example of a signal deactivation. Phosphatases are going to remove phosphate groups from proteins in the phosphorylation cascade. Such a mechanism is going to allow the cell to remain sensitive to small changes in signaling. Now let's talk about crosstalk. So crosstalk is how we're going to integrate diverse signals that a cell can receive, right? A cell likes to talk to a lot of people at a time, right? We're not only going to receive one signal. We have to be able to make sense of all these different signals happening at once. So there's three important ideas here. One pathway may inhibit steps in a second pathway, and that could ultimately reduce the cell's response to the first pathway. We spoke a little bit about this before when we mentioned things like negative feedback inhibition. One pathway may stimulate the steps in a second pathway. This would be like an activation step, and could ultimately lead to two different responses to a signal. Multiple steps in a signaling pathway provide many points for crosstalk to occur. Right? Let's say I have a giant metabolic pathway from steps A to C to Z. Step A may activate step C, but step C may deactivate the impact that Z would have. So you have to remember that all these pathways are cross-talking and interacting with one another. Alright, so here's like a giant pathway, right? Our cell membrane, multiple receptors, different receptor responding to a different signaling molecule. Pathway A for signaling molecule A, pathway B for signaling molecule B, and so forth. So during crosstalk, what's going to happen? Let's say A binds to our receptor, we generate some cascade. Intermediate A1 may affect receptor B. It can cause receptor B to not function, right? It may change its shape, so now signaling molecule B cannot bind. Therefore, if signaling molecule B does not bind, we don't get the cell response to B. So this is what crosstalk means. Each signaling pathway is for a specific receptor hormone interaction. However, the intermediates of one pathway can either inhibit or stimulate the intermediates of another pathway. Okay, so even unicellular organisms have to be able to respond to their environment and communicate with one another. Often, the signaling communication between these unicellular organisms is going to be based on their environment, particularly density. So, signaling pathways that respond to a population density is known as quorum sensing. Bacteria release species-specific signaling molecules when their numbers reach a specific threshold. In response, they're going to release signaling molecules, and these signaling molecules are going to vary across species. Some examples of quorum sensing would be development of plaque on your teeth. So bacteria are going to use quorum sensing to help glue a community of microbes to form a biofilm. Okay, signal reception. So signal receptors are dynamic. The number of receptors present in a cell or on a cell membrane can change. The ability of a receptor to bind a hormone can also change. Overall, sensitivity of a cell to a particular hormone can be modified over time. Also, signal receptors can be blocked. So just understand that we can only have cell signaling if we have the receptor present. A lot of different things can happen to our receptor. We may have another protein that binds to it, right? Maybe it's phosphorylated and it alters its shape and it no longer can bind to its signal. If we can't bind to our signal, we cannot transduce an extracellular to intracellular message.